So first of all, welcome. Thank you so much for participating. We're excited to talk to you today. Yes, thank you. First question, please share some background information on Prodigest and its role in the prebiotic space.
Yeah, Prodigest has been founded in 2008. We are a contract research organization specialized in simulation of the physiology and microbiology of the gastrointestinal tract of humans and of animals. Our mission is simple. We make our clients successful by de-risking their product development. And we do that by providing mechanistic insights on the functionality of their ingredients using cutting-edge technologies to simulate the gut with proven in vitro, in vivo correlation. Our core technology is the SHIME, which stands for Simulator of the Human Intestinal Microbial Ecosystem. And this is a computer-controlled device that recreates the physiology and microbiology of the different areas of the digestive tract. In the most advanced configuration and application, you can use such a platform to perform a clinical trial in vitro with a schedule of several weeks. Prodigest has at the moment 41 full-time employees, of which 10 PhDs with at least 7 years of relevant experience in the sector, labs and offices for 1,600 square meters, and we are an official biobank since 2020. Prebiotic space has been the first area of business for Prodigest after its incorporation, and it remains a very important area of action for us, representing a good percentage of our yearly turnover.
Great, thank you so much. So can you elaborate on the research and development efforts your company invests in to advance the understanding and application of prebiotics in promoting health and well-being?
Yeah, innovation is at the base of our DNA because we constantly strive to improve our technologies, our protocols, our QC procedures to deliver the best scientific data to our clients and to retain a position of leadership in the market. Every year, approximately 15% of our turnover is invested in R&D, and this led to the development of the most complete pipeline of in vitro tools in the market to simulate the GI tract of humans and animals under health and disease conditions for different age categories, from babies to elderly, and from high throughput to detailed mechanism of action. So essentially, I can say we are the partner of choice to replace, refine, and reduce the use of animals in gut research, and to test the effect of prebiotic against a condition that truly represents the target population of choice.
That's really interesting. I'm familiar with what you all do, and it's interesting to hear it from you, diving deeper into these kind of questions. How does your company stay abreast of advancements in gastrointestinal research and technology to continuously enhance capabilities of its technologies?
Yeah, in this field, our approach is quite simple. We are constantly in contact with our clients and we learn from them about their needs for the near future and for the years to come. So I think this is the best possible approach as we evolve in a direction that we know will be able to support the research questions of tomorrow from our clients. We also make use of client satisfaction forms after every project, for which I have to say the score is quite very high. And from there, from these forms, we are able to derive useful information for future developments. Last but not least, being part of an association such as, for instance, GPA helps, again, in having a good view on the current and future challenges for the field. So all of this guide then our R&D efforts.
That's awesome. Thank you. Can you discuss any recent innovations or breakthroughs in the prebiotic technology that Prodigest has been involved in and how the advancements differentiate your position in the market?
Yeah, the market is moving in the direction of precision nutrition and precision medicine. This implies that lead products must be tested under conditions which are representative for the target population and also taking into account the inherent intra-individual variability. The result is that projects nowadays require the capacity of testing tens to hundreds of conditions in parallel. So with this in mind, in 2020, we developed and validated our colon on a plate technology, which is a high throughput ex vivo simulation of the colon. Moreover, another important thing is that when you generate hundreds of samples to analyze, you also need a high throughput analytical tool to then control the budget. For this purpose, we make use of our laser-based mass spectrometry, it's called LA-RAMS, which allows to obtain an untargeted metabolic fingerprinting of 400 samples per day at a very low price. And I'm happy to say that the combination of colony plate and LA-RAMS is rapidly becoming a standard tool in the prebiotic field. And this combination opens the possibility of addressing research questions based sometimes on hundreds or in a couple of projects, even thousands of comparisons.
That's interesting. And then are there any recent partnerships or collaborations your company's established with industry players or research institutions to further advance the industry?
Well, it is difficult to pinpoint a specific project out of many, because in the past 15 years, we collaborated with more than 500 clients worldwide, some of them recurrent, and we supported more than 2,000 projects. So the results from research involving our technology platform conducted directly by us or by our partners ended up in more than 300 scientific papers. So in practice, it's difficult to say one project, but we are happy of the opposite. In fact, we are very proud of the fact that with our expertise, we can contribute to the development of multiple innovative products. And this is frequently the case in science, in which every advancement is the results of many years of effort. Is the famous statement of Isaac Newton: if I've seen further, it is by standing on the shoulders of giants. And basically, this is what we try to do every day. So yeah, coming back to your question, I'm not able to pinpoint a specific collaboration, but on the contrary, I can say that we can actually make a constant effort to deliver top science to our clients. And in this way, we provide a constant contribution to push forward the boundaries of science in the prebiotic field, but not only in the prebiotic field.
Great, thank you. And what health areas are most interesting for the work that your team is doing, and what aspects? For example, is it MOA? Is it SCFAs, other metabolites?
Yeah, the focus of our work is always gut related. So gut health and immune modulation by coupling, for instance, gut simulation together with cell assays still cover great part of our research. However, more work is nowadays requested in terms of gut-organ connection, as for instance, gut-brain or gut-skin, and also to assess the impact on specific disease states, such as IBD or type 2 diabetes or Parkinson. So these are very interesting requests that frequently moves the investigation from the classical short-chain fatty acids as a result of saccharolytic fermentation to other metabolites. And for this purpose, we make use of our MetaKey platform, which is a metabolomic platform composed of a laser-based mass spectrometry, the one I already mentioned before, and then the classical LCMS approach. By developing specific panels of metabolites, we can assess the potential impact of the treatment on a specific disease condition or on other organs in the body in an indirect way. For instance, we have a neuroactive panel or a panel for tryptophan metabolism or bile acids, just to mention some. There are other two areas that I can also mention of strong interest. One is about the development of symbiotics, therefore the use of the combination of probiotics and prebiotics, and also the use of prebiotics for pet application, for which our models have been adapted to simulate the dog and the cat gastrointestinal tract.
Interesting. And finally, our last question, can you share any case studies or success stories where your simulator-based testing has led to significant advancements or innovations in the development of prebiotic products?
Yeah, leaving aside the different cases which are open to the public because there is a publication or there is a press release or an announcement in LinkedIn, these questions brings me to my mind a specific studies that shows the full power of our pipeline. At Prodigest, we strongly believe that it doesn't exist the single model that fits all the purposes. In fact, the selection of the model depends on the research question. So in this specific project that I was mentioning, we had to test multiple lead products to assess the possible structure-effect correlation with multiple donors. So at the beginning of the project, because we have to deal with a lot of conditions, we made use of our column and plate approach to screen all the different leads compounds against the microbiota of 9 different donors. So a total of experiments of around 200 simulations. We then made use of our metabolic fingerprinting to further fine-tune the effect of the products, not just focusing on simple short-chain fatty acids. This led to the selection of some of the test products in a sort of a funnel approach. Next part of the work had to answer questions related to sustained fermentation along the column, proximal versus distal column, for instance, temporal onset of the effect, especially for those fibers with a high degree of polymerization and a high degree of substitutions. For this type of question, a short-term simulation is not fit for purpose. So we made use of our SHIME to assess the effect of daily repeated doses of the selected prebiotics. The research then resulted in a definition of a specific combination of prebiotics that could induce a sustained fermentation all along the column. So in a nutshell, a project that shows that you need the right technology and you have to use it to provide the right scientific answer for that.
Great. Thank you so much, Massimo, for diving into all of the things that Prodigest, brings to the prebiotic space. We've really gotten, a lot out of getting to know you guys a little bit better and diving into, the technical stuff a little bit deeper on what sets you guys apart. So thank you so much.
Thank you, Morgan, again, and thank you for the opportunity.